Hello everyone. Today we are going to understand transcription process. Already DNA replication has been taught to you. So let's begin. Okay, so first let's see a very important phenomenon called as central dogma. Now what is actually central dogma, one might ask? It means a simple process where DNA gets converted to RNA and RNA gets converted to proteins. Last lecture, when we were studying, we have understood one important parameter that DNA can replicate itself and it requires an enzyme called as DNA polymerase. Okay, so when you want to replicate DNA from DNA strand, it is self-replication and you require DNA polymerase. DNA to RNA, you require something called as RNA polymerase. So what is this polymerase name actually given on? What is to be produced? So when DNA has to be produced, it's DNA polymerase. When RNA needs to be produced, it is RNA polymerase. Okay. And then from RNA to protein. Okay. Now over here, there are different names to this process. DNA to RNA is called transcription, whereas RNA to proteins is called translation. This is the second part of the series of molecular biology where we are studying about transcription. Okay, so we are basically going to understand production of different types of RNAs. Okay, so let's begin. While understanding this, let's take a simple example over here. Let's say there are two DNA strands, strand 1, strand 2. About DNA polarity, we have already discussed, right? So let's say strand 1 is running 5' to 3'. Strand 2 is running 5' to 3'. So DNA is basically a double-stranded structure and the two strands are complementary to each other and anti-parallel, meaning they are opposite in their polarity. Let's take some codons over here. A, T, G, T, C. Okay. So what would be the complementarity over here? It would be T, A, C, E, G. Now, for those who don't know about this or are hearing it for the first time, always remember there are 4 nucleotides, nucleotide bases: adenine, thymine, guanine, guanosine, and cytosine. Okay, so there are 4 nitrogenous bases. There's another nitrogenous base called as uracil, which is only found in RNA. Okay, so adenine binds to thymine using a double bond. Guanine binds to cytosine using a triple bond. Okay, so that's why wherever adenine was placed, I have written thymine in the complementary strand. Wherever guanine is placed, I am writing cytosine in the complementary strand. Okay, so now let's see if suppose mRNA is made from this. What would happen? mRNA would be made in this direction. Are we clear with this? It would be made in this direction and it would be 5', 3'. Okay. So as I've told you, uracil is only found in RNAs, right? So in RNA, there is no such thing called as thymine. Instead of thymine, we have a base called uracil. So adenine is going to code for uracil over here. Okay. T is A, G is C, T is A again, This is G. Okay. So what have we done? First things first, we need to understand that these two, 5' to 3' in black, are the DNA. Okay, the DNA that we are using to form mRNA is the template strand or template DNA. Okay, this is your mRNA or RNA. The strand that is not utilized for making of mRNA is called as complementary strand or complementary DNA. Okay, what else can we talk about this? Can you see over here? Except for uracil and thymine, adenine, adenine, cytosine, cytosine, adenine, adenine, guanine, guanine. What does this indicate? This indicates that mRNA is almost similar to one of the strands in DNA. So complementary strand is also called as coding strand, coding strand, or sense strand. Now one might say it is not the strand that was used to make RNA, still it is called as coding. It's because your RNA is almost similar to your complementary DNA. We always need to know one thing, that template DNA and complementary DNA are nothing but complementary to each other, right? So they were wound up, we opened them, and one strand we took for making RNA. So there is a good chance that the RNA would be similar to one of the DNA strands. So how I help my students remember, you can even call template strand as antisense because it is complementary or opposite. How I help my students remember this is using a simple notion that template strand is a strand which makes RNA, and complementary strand is a strand which is copy of RNA or copy RNA. You can also write it as similar to RNA. No problem. Okay. So one which is making RNA is going to be your template strand, and one which is similar to RNA is going to be your coding, complementary, or sense strand. These things are very important to remember and they form the base of the chapter. Many people ignore this and later on they get confused when coding strands are used, when reference to template strands are used. People definitely get confused. So I would ask you to pause for 5 minutes, understand this, interpret it properly, and then move ahead. Okay. Now, the question that most famously is asked is why both the DNA strands, why both the DNA strands not used to make RNA. Okay, now we know that both are complementary. So why not use both the strands of DNA to make RNA? Always remember that complementary does not mean same. Okay, whenever we are making proteins, always remember this. Transcription is copying DNA to RNA. Translation is decoding into proteins. Proteins are important, right? And proteins work as codons, triplets. So ATG, they code for one particular type of amino acid. You just see the complementary CAT, TAC, GTA. These are different forms. Okay, they are not the same thing. So when I'm going from 5 prime to 3 prime, let's say for example, I'm reading over here ATG. This ATG would code for an extremely different amino acid when compared to CAT, right? So you are not going to get the same amino acids, even though it's from the same DNA. Okay. Complementarity does not equate to similarity. It is only giving you this relation, nothing else. Okay, yes, mRNA, one mRNA and coding strand are definitely the same, but template strand and complementary strand are not the same. The arrangement of the triplet codons are exactly not the same. They are going to code for different amino acids, make different proteins, and that would be problematic. Okay, I hope you have understood this much. So first thing first, why that thing can't be done, why both the strands can't be used. First reason would be that both DNA are complementary. Both DNA strands are complementary. This is a very famous question asked, and a lot of people get confused. Both strands complementary. They are not the same. Complementary. Not same. So they'll end up coding different proteins. Okay, they'll end up coding different proteins, and that would be problematic. Second reason why, if two RNAs are made, two RNAs made from two DNA strands, from complementary DNA strands, what would happen? These two RNA, because they are made from complementary strands, would end up being complementary for each other and will form double-stranded RNA. And always remember, only single-stranded RNA can help code proteins. Translation will occur only when the RNAs are single-stranded. In double-stranded RNAs, there would be no coding for proteins. Translational activity does not occur with double-stranded RNA. So the two reasons are here mentioned, and that is the reason why things are a bit dicey when we talk about transcription. That therefore I started with explaining this concept first, so it becomes crystal clear on what we are taking into consideration. Now, once we have understood the base of transcription, let's try to understand a very important phenomenon called as transcription unit. Called as transcription unit. Now, this transcription unit would be having three things. There would be one promoter. If you have gone through my Lacoperon system, you might have understood what are promoters, what are terminators. So please refer them for understanding the detail. Then there are some structural genes which actually code for some proteins. And finally, there are terminators. So the ones that give rise to the actual genes or actual proteins in our body are nothing but structural genes. So they are important. Promoters and terminators help us locate and terminate the transcription of the genes. Okay, they are like the start and end signal. So promoter is nothing but the start signal. Okay, it gives you a red sign, sorry, it gives you a green sign, my bad, to start the transcription, and terminators are like the red signals which give the end sign to stop the what we can say transcription. Now let's see how these are arranged. Let's say, for example, we have got our two strands running over here. We have one 5 prime to 3 prime. Another one is 5 prime to 3 prime. Okay. Now, in the last slide itself, we have understood there would be one template strand and there would be one coding strand. How I personally feel one should remember promoter and terminator is with respect to coding strand. Now, how does your terminator and promoter fix itself? Here comes your promoter. Here comes your terminator. Okay. So here is your promoter. Here is your terminator. So you can say, where is the location of promoter? Promoter is where it's on the 3 prime end of template strand. Okay. Or it's at the 5 prime end of coding strand. So you can see over here, right? You can give the position of promoter with respect to both the strand. But whichever strand you are mentioning, mention it in detail. Do not just write promoter is at 3 prime end or promoter is at 5 prime end. Of what? Which strand? Because if you don't clarify, there is going to be a huge doubt. Okay. Next, terminator can be given as terminator is at the 5 prime end, okay, of template strand or is at the 3 prime end of coding strand. So you can position them, label them according to your wish, but please make sure to properly allocate the strand name along with it. That would be helpful. There is a general perception of naming the position or locating the position of promoter and terminator with respect to coding strand. So how do we actually take it? Okay, this happens to be your structural gene area. Okay, this is where the structural gene is going to be present. So with respect to coding strand, we can say promoter is 5 prime upstream of the coding gene. Upstream means before, whereas terminator is 3 prime downstream of structural gene. What are we referring to? We are referring to coding strand. So generally there is a perception of positioning your promoter and terminator with respect to the coding strand, right? Coding strand is the one which is not utilized to make the RNA. Rather, it is similar to RNA. Okay. So either which way you can name it, locate it, but make sure this thing stays in your mind, how to annotate it. Once you have understood this logic, let's go ahead. Let's try to understand things from a different perspective. Okay. Next thing that we can learn for is structural genes. Okay. We know one thing, that structural genes can be okay. Structural genes can be positioned in two ways. Let's say there is one promoter, there is one structural gene, and there is one terminator. Another scenario could be there is one promoter, there are multiple structural genes, and there is one common terminator. Okay. Looking at the second diagram, second chart, you might have been referring yourself to one amazing parameter we have discussed, that is operon, right? So example is operon, where multiple structural genes are present between one single promoter and terminator. Whereas most of the eukaryotic genes, most of the eukaryotic genes have only one structural gene, and every structural gene will be represented with one promoter and one terminator. So it's pretty much like, an individual feature, whereas in prokaryotes, one promoter and one terminator can be used for multiple genes aligning together. Okay, so such a feature where one structural gene is guarded by a promoter and one terminator individually, it's called as monocistronic gene. It's called as monocistronic gene. Whereas whenever multiple structural genes are guarded by singular promoter and terminator, these are called as polycistronic genes. Okay, this is kind of an important feature. And if you have not gone through the lacoperon video that I had uploaded, make sure you go through it. You'll get much more highlight, and that too in detail. Okay, that was about structural gene. Now let's dive into the major juice of transcription. Okay, so in transcription, there are two important terms which you cannot miss if you want to understand transcription with respect to prokaryote and eukaryote. The two important terms are what? They are introns and exons. Okay, so every time your transcription is going to happen, there is going to be an RNA which is produced. That RNA mostly has got two segments, okay, fragmented with each other. These two segments are introns and exons. So what are actually the introns? Introns are that part of the RNA which do not get translated to proteins. Okay, they do not get translated to proteins. Okay, they are just present in the RNA to increase the girth, I feel. Okay, and also another thing you need to remember is they are not present. They are not present. You can even write this as absent, otherwise you might get confused. They are absent in mature mRNA or mature RNA. Okay, so introns just increase the space area of the RNAs. They are not actually coding for any proteins. And while you're going to process the RNA into a mature one, you're going to remove the introns. So introns are segments of genes which do not code for any proteins and are removed from the mature RNA. Exons are just the opposite. Exons are the ones which are translated to proteins. So they are the segments of RNA that get translated to proteins. Okay. And because they are going to be translated to proteins, they are going to be present in mature RNA. Okay, these were a couple of terms that we need to remember, couple of concepts that we remember before diving into transcription process. Now, transcription process is basically divided into three parts. We all know about it. Transcription has got three parts. One part is initiation. Okay. Second part is elongation. And third part is termination and related processing. Okay, so these are the three steps of transcription. Prokaryotic and eukaryotic transcriptions work in a different manner. Let's try to understand prokaryotics. Okay, what happens in prokaryotic transcription? Over here, always remember one important thing. In prokaryotes, there are not going to be any introns, meaning whatever is coming out in the mRNA or RNA, that is going to be only exons. You're not going to have any unnecessary segments. The reason being, prokaryotic DNA is very much concise and conserved. See, these organisms are pretty unicellular or small or minimalist, right? They have all their functions piled up in one cell most of the time. Unlike eukaryotic organisms where functions get divided in different cells, prokaryotic organisms carry out all the functions in one cell. So they are all about conservation of space, right? So there are no unnecessary genes present. All the things, your DNA, if we talk about, see over here, let's take this to be our DNA. Okay, and we have another part over here. Okay, let's term this as 3 prime, 5 prime, and let's term this strand as 5 prime, 3 prime. Where does the promoter come in bind? Promoter comes in binds on what? It comes in binds either on 3 prime of your template or 5 prime of your complementary or coding. So for this, I feel whenever you're writing it down, write down this. First, green one is your template and the black one is the coding. So that might make things easier for you. Okay. So here we would be having a promoter. Okay. And somewhere down, we might be having a terminator. Now these promoters and terminators, they do not travel on their own. They have certain factors which are going to help them travel. So for initiation in prokaryotes, there is a factor or protein called as sigma factor. This sigma factor helps RNA polymerase to come and attach itself or locate itself to the DNA strand. Okay, so sigma factor, let me name this for you. This is sigma factor. Sigma factor helps RNA polymerase come and attach to the template strand. Once RNA polymerase attaches, it goes ahead, okay, it opens up a DNA a little bit and starts transcribing your mRNA. This is called as elongation. Your mRNA comes out in the form 5 prime and the one that is being synthesized is 3 prime. So this is how your mRNA starts coming out. Okay. And finally, when your work is complete, when transcription has completed, that time another factor called as rho comes and attaches to the polymerase. Okay, and it says that yes, polymerase, now your work is over. We are at the terminator. You need to leave. What is this factor? This is rho factor. And it helps the RNA polymerase leave or terminate the process of transcription. So stepwise, if we want to know, in prokaryotes, there are 3 steps. Step number 1, step number 2, and 3. First step is initiation, that is bringing in the polymerase, RNA polymerase. And this initiation is done by a factor called as sigma. Sigma does what? It brings RNA polymerase to promoter. Okay. Second step is going to be elongation. And over here, your mRNA is going to be transcribed. Okay, it's coming out. Next one is going to be termination, the final step. In termination, there is a factor called as rho, and rho detaches. Rho does what? It detaches RNA polymerase at terminator. So these are the three steps that we need to remember. Rather, in your prokaryotic transcription, things are rather simple. There are going to be only exons, so there is no need of processing. No further processing to remove any unnecessary intron, right? So there is no further processing. Because there is no further processing, what is going to happen? The transcription would be simultaneously done with translation. See, there is no other processing required. So your whatever mRNA or RNA is coming out from transcription is already mature, properly made. So as soon as the RNA is coming out, translation process, that is production of proteins, keeps on happening simultaneously. Prokaryotic transcription happens where it is going to happen in cytoplasm. So both transcription as well as translation of prokaryotes happens in what? Cytoplasm. Reason being, there is a nucleoid structure, no definite nucleus for DNA to sit in. So DNA is partially suspended in cytoplasm, right? So what is prokaryotic transcription? Let's go through it. There are no introns, there are only exons. So as prokaryotic transcription is happening, mature RNA is coming out, which gets translated to protein. So first, at the 3' end of the template or 5' end of the coding, a recognition factor called as sigma brings in the RNA polymerase to promoter. Okay, brings in the RNA polymerase to promoter. And then the process of transcription will start where DNA would be converted to RNA. Okay, it would be copied into RNA, and RNA polymerase would be one by one leaving the nucleotides out. Next, what is going to happen at the terminator region? There is a particular protein called as Rho protein or Rho factor, which is going to tell or signal the RNA polymerase that the transcription of the structural gene has been completed. Now you may leave. So at that point, when Rho recognizes terminator, it is going to detach the RNA polymerase. Even if you remember this much, more than sufficient, no need to dive into any other concepts regarding. Okay. Our eukaryotic transcription works a little bit different because of the complexities. Let's move on to eukaryotic transcription. Eukaryotic transcription is a little bit complex. Let's see how things are going to work. There is a reason for it. The reason being that eukaryotic transcription has got your, what we can say, introns as well as exons, right? So let's see how things work. Let's say this is 3', 5'. Over here we have 5', 3'. Let's take this to be template, the red one. Let's take the black one to be coding. Now, mind you, eukaryotic transcription takes place in the nucleus where the DNA is present. Eukaryotes have a definitive nucleus. Our DNA is not suspended in the cytoplasm. Okay, so what is going to happen? Over here, we will be having promoter region. terminator region, correct? Okay, so we would be having a promoter over here. Not bad. We would be having a promoter over here. And we would be having a terminator over here. Now, to the promoter, there are certain factors called as PIC. PIC is what? It is pre-initiation complex. Okay, there are many transcription factors, okay, n number of them which come together for recognition of promoter and terminator. So you have not been given the exact names or number of PIC which is joining, but this PIC is the one which will help in bringing RNA polymerase. Now thing starts over here. RNA polymerase in eukaryotes are of three types. Okay, there are three RNA polymerases, RNA polymerase 1, 2, and 3, right? RNA polymerase 1 generally codes for your rRNA or ribosomal RNA. Okay, RNA 2, polymerase 2, codes generally for heterogeneous RNA, okay, which will be further matured into your mRNA. We'll be learning about this step in a couple of minutes. And RNA polymerase 3 actually codes for tRNAs, pretty important in protein production, and also small nuclear RNAs. Okay, so unlike prokaryotic transcription where there was only one common generic RNA polymerase, eukaryotic transcription rather has got three polymerases working, are polymerase 1, 2, and 3. Okay, now what is going to happen? This hnRNA, how does an hnRNA look like? Let's see. Heterogeneous RNA would be having smaller chunks. What would be having? Smaller chunks of your introns. Okay, these are smaller chunks of introns. And rest of the parts would be exons. I have told you in prokaryotes, there are going to be certain introns. Okay, and there are going to be certain exons. Now, this is your hnRNA, heterogeneous RNA. Here starts our first step. First step of processing is what? First step says is adding methylated GTP to the 5' end of hnRNA. Okay, so what is going to happen? This is our 5' end with our introns intact, and over here at the 5' end, there is going to be a GTP molecule with a methylated group, methyl group, okay, which will be attached. Fine. So these are introns and introns are still present. Second step is called as splicing. Second step is called as splicing. This step is also called as capping. First step is capping. Second step is splicing. Now in splicing, what is going to happen? You are going to remove introns. Okay, so all the introns are removed. Now your mRNA is pretty clear. It has only exons in it. Okay, it has only exons in it. And the third step is going to be tailing. Now tailing is also called as polyadenylation step, where what is going to be happening? Your now so mature RNA will be having at 5' end a GTP molecule which is methylated. It would be having only exons which are coding for proteins, and at the 3' end it would be having a poly A tail. Okay, poly A tail. What is the reason for these things to happen? All these things, capping, tailing, they are required for one process. In nucleus, there are something called as endo and exonucleases, which are very much notorious. They cleave any single stranded particles which are just roaming about. To protect our mRNA, which is now mature, from these nucleases or enzymes that break down, we require capping and tailing process. So it is just like a security guard, Z plus security. Remember, eukaryotic transcription happens in nucleus. The mature mRNA then goes to the cytoplasm for production of proteins. And this is how the entire concept of transcription start to end is completed in this video. If you have already understood transcription, please make sure to watch this video at 2x and revise every time. Signing off till the next time. Bye everyone.